Unit 4, Biotechnology 1. The term biotechnology was coined by Carl Irecki. 2 restriction enzymes were discovered by Arbor, Smith, and Nathan. 3. The first restriction endonuclease discovered was Hind II. 4. Restriction enzymes cut DNA at specific sites called recognition sites. 5. The enzymes that join DNA fragments are called ligases. 6. The enzyme reverse transcriptase was discovered in retroviruses. 7. Plasmids are used as vectors in genetic engineering. 8. T plasmid is found in Agrobacterium tumefaciens. 9. The process of introducing foreign DNA into host cell is called transformation. 10. PCR stands for polymerase chain reaction. 11. The scientist who invented PCR technique was Kary Mullis. 12. The thermostable DNA polymerase used in PCR is Taq polymerase. 13. Electrophoresis separates DNA fragments according to their size. 14. DNA is negatively charged due to phosphate groups. 15. Agarose gel is obtained from seaweeds. 16. The process of making multiple copies of DNA is called DNA amplification. 17. The technique of making identical copies of an organism is called cloning. 18. The first cloned mammal was Dolly sheep. 19. Dolly was cloned from a cell of mammary gland. 20. The introduction of recombinant DNA into host cells is called transfection. 21. The protein crystals of Bt toxin are produced by Bacillus thuringiensis. 22. Bt cotton is resistant to insects. 23. RNA technology is used to silence the expression of a gene. 24. The first transgenic cow was Rosie. 25. Rosie produced milk rich in human protein alpha-lactalbumin. 26. Insulin used for diabetes treatment is produced by recombinant DNA technology. 27. The 2 chains of insulin are connected by disulfide bonds. 28. The first clinical gene therapy was done for ADA deficiency. 29. Golden rice is rich in vitamin A30. Biopat means protection of biological resources or products. 31. Biopiracy is the exploitation of bioresources without authorization. 32. Transgenic plants resistant to pests are called GM crops. 33. Flavor Savor tomato was genetically modified to increase shelf life. 34. The enzyme used in gene therapy to cut DNA is restriction endonuclease. 35. Somatic gene therapy affects only individual, not offspring. 36. Cry genes are responsible for insecticidal proteins in Bt. 37. Apiculture is the rearing of honey bees. 38. The first recombinant DNA molecule was constructed by Cohen and Boyer. 39. DNA fingerprinting technique was developed by Alec Jeffreys. 40. Bioethics deals with moral issues in biotechnology. 41. The term biotechnology was coined by Carl Irecki. 42. Restriction enzymes are also called molecular scissors. 43. The first restriction enzyme was discovered by Smith and Nathans. 44. The enzymes that join DNA fragments are called ligases. 45. The enzyme reverse transcriptase synthesizes DNA from RNA. 46. The vector widely used in plant transformation is T plasmid. 47. The commonly used cloning vector in E. coli is PBR32248. The small circular DNA in bacteria is called plasmid. 49. The enzyme Taq polymerase is obtained from Thermus aquaticus. 50. Taq polymerase is used in PCR. 51. PCR stands for polymerase chain reaction. 52. The machine used in PCR is called thermocycler. 53. The process of separating DNA fragments by charge is called gel electrophoresis. 54. The gel commonly used in electrophoresis is agarose. 55. DNA fragments are visualized using ethidium bromide. 56. A blotting technique used for DNA is called southern blotting. 57. Northern blotting is used for RNA. 58. Western blotting is used for proteins. 59. The first recombinant DNA was made by Stanley Cohen and Herbert Boyer. 60. The first transgenic plant was tobacco. 61. The first transgenic animal was a mouse. 62. The first transgenic cow was Rosie. 63. Rossi produced milk rich in alpha-lactalbumin. 64. Human insulin produced by recombinant DNA is called humulin. 65. The technique of making multiple identical copies is called cloning. 66. The first cloned mammal was Dolly the sheep. 67. The technique used to clone Dolly was somatic cell nuclear transfer (SCNT). 68. Golden rice is rich in vitamin A. 69. Bt cotton is resistant to insects. 70. The gene for Bt toxin is obtained from Bacillus thuringiensis. 71. RNA stands for RNA interference. 72. RNA is used for gene silencing. 73. The patenting of biological products is called biopat. 74. GEAC stands for Genetic Engineering Approval Committee. 75. The process of treating waste with microbes is called bioremediation. 76. The microbes used for cleaning oil spills are called Pseudomonas. 77. Microbes used for alcohol production are yeast. 78. Microbes used for antibiotics are fungi and bacteria. 79. Cyclosporin A is produced by Trichoderma polysporum. 80. Statins are produced by Monescus purpureus. 81. Genetically engineered bacteria are used to produce insulin. 82. The first clinical gene therapy was done for ADA deficiency. 83. ADA stands for adenosine deaminase. 84. The first gene therapy was given in the year 1990. 85. Gene therapy can be of two types, somatic and germline. 86. Stem cells can be used in tissue repair. 87. The full form of GMO is genetically modified organism. 88. The organization responsible for safety of GMOs in India is GEAC. 89. Cryproteins are toxic to insects. 90. Bt cotton is resistant to bollworm. 91. Golden rice is genetically enriched with beta-carotene. 92. Bioethics deals with ethical issues in biology. 93. Biopiracy refers to illegal use of biodiversity. 94. Biopat refers to patenting of biological products. 95. DNA fingerprinting is based on VNTRs. 96. The scientist who developed DNA fingerprinting is Alec Jeffreys. 97. In India, DNA fingerprinting was introduced by Lalji Singh. 98. DNA fingerprinting is used in paternity disputes. 99. DNA probes are used in diagnosis. 100. ELISA test is used to detect AIDS. 101. Recombinant DNA technology was developed in 1973. 102. The bacterium used in Bt cotton is Bacillus thuringiensis. 103. The nematode-resistant plant was developed using RNA interference. 104. The nematode that infects tobacco plants is Melodogene incognita. 105. Gene silencing occurs through dsRNA. 106. The first animal cloned was Dolly. 107. Dolly was cloned from the cell of mammary gland. 108. SCNT stands for somatic cell nuclear transfer. 109. Cloning is a method of producing identical copies. 110. Genomic library contains entire genome sequences. 111. cDNA library contains only expressed genes. 112. The fluorescent dye used to stain DNA is ethidium bromide. 113. The enzyme used to cut DNA is called restriction endonuclease. 114. The palindromic sequences are recognized by restriction enzymes. 115. A plasmid that carries foreign DNA into host is called a vector. 116. The process of introducing DNA into host cell is called transformation. 117. Biolistic method is also called gene gun method. 118. Agrobacterium tumefaciens causes crown gall disease in plants. 119. The enzyme used to seal DNA fragments is DNA ligase. 120. The technique of DNA amplification in vitro is called PCR. 121. The full form of PCR is polymerase chain reaction. 122. PCR was invented by Kary Mullies. 123. Taq polymerase is isolated from Thermus aquaticus. 124. The cutting enzymes in biotechnology are called restriction endonucleases. 125. E. coli produces sticky ends. 126. Restriction enzymes were discovered by Werner Arber. 127. The DNA molecule used to transfer foreign DNA is called vector. 128. Plasmids are commonly used as cloning vectors. 129. The first artificial recombinant DNA was created by Stanley Cohen and Herbert Boyer. 130. The connecting enzyme used in rDNA technology is DNA ligase. 131. The process of separating DNA fragments is called gel electrophoresis. 132. The commonly used gel in electrophoresis is agarose gel. 133. In agarose gel electrophoresis, DNA fragments move towards the positive electrode. 134. The process of transferring DNA fragments onto a nitrocellulose membrane is called southern blotting. 135. DNA fingerprinting was developed by Alec Jeffreys. 136. The commonly used selectable marker in vectors is antibiotic resistance gene. 137. Bioreactors are used for large-scale production of proteins. 138. The main advantage of bioreactor is continuous culture. 139. Bt toxin is produced by Bacillus thuringiensis. 140. The gene for Bt toxin is inserted into crops to provide insect resistance. 141. The first transgenic cow was Rosie. 142. Rosie's milk contained human alpha-lactalbumin. 143. The first transgenic plant produced was tobacco. 144. Insulin is produced by inserting human insulin gene into E. coli. 145. The first genetically engineered human insulin was named Humulin. 146. Gene therapy was first used for ADA deficiency. 147. The first human to be treated by gene therapy was Ashanti da Silva. 148. CRISPR-Cas9 is a tool for gene editing. 149. Agrobacterium tumefaciens transfers genes through its T plasmid. 150. Golden rice is an example of biofortified crop. 151. VNTRs stand for variable number of tandem repeats. 152. VNTRs are useful in DNA fingerprinting. 153. The first cloned sheep was Dolly. 154. Dolly was cloned by Ian Wilmot. 155. Dolly was produced by technique called somatic cell nuclear transfer. 156. Gene silencing using RNA interference was first discovered in tobacco plant. 157. In RNA, the double-stranded RNA binds to mRNA. 158. The RNA that interferes with gene expression is called cRNA. 159. Biopatent is a legal right granted to an innovator. 160. Biopiracy is the use of biological resources without authorization. 161. In electrophoresis, smaller DNA fragments move faster. 162. The full form of GMO is genetically modified organism. 163. The first GM crop released for commercial use was Flavor Savor tomato. 164. Pseudomonas putida is used to clear oil spills. 165. The bacteria used in gobar gas plants are methanogens. 166. The most abundant greenhouse gas is CO2. 167. The most potent greenhouse gas is CFC. 168. The full form of IPCC is Intergovernmental Panel on Climate Change.